Right, here's question 1 from section 2 of the National 5 biology exam from SQA in 2023. And this question is about cell biology, so we'll have it together with other cell biology questions and you'll be able to see the method for solving this question. Let's read the question, okay? So palisade mesophyll cells are found in leaves and carry out photosynthesis. The diagram shows a layer of these cells viewed under a microscope. Now, they're called the palisade mesophyll cells because they're aligned vertically, right, so that they can catch the sunlight coming straight down onto the leaf. Okay. Above them, there would be some epidermal cells and a cuticle of some kind. And then below them, you'll have the spongy mesophyll. And below that, you'll have the guard cells. And the spongy mesophyll will allow carbon dioxide to diffuse into these cells. Okay, so we've had a look at the picture. This is an image from a microscope, so a circular image of a light microscope. And that seems a reasonable enough magnification. Okay, so what information are we given? We're given a letter P pointing to an object in the cell. And then we have a total distance across the field of view is 0.25 millimeters. And it says here that 1 millimeter equals 1,000 micrometers. So that would mean, I think just doing a calculation in my head, that that's 250 micrometers across this field of view, 250 micrometers. Okay, so let's see what the questions are. It says here, name structure P. Well, we'll go up here, and if you can remember all your drawings from biology, this is the double line that we use to represent a cell wall, and they use it in the exam. So that is a cell wall. Always try and write neatly. Try not to make too much of a mess, but if you make a mistake, don't waste your time whiting out or anything like that. Just put a line through it and write the correct answer, or your preferred version in the end. May not be correct, who knows? Depends how well you're thinking on the day. So calculate the average width of a single palisade mesophyll cell in micrometers. Well, we've got the measurement in millimeters. We need to convert to micrometers first, or we need to do the calculation and then convert to micrometers, because we've got to get this answer in micrometers. They've given you the unit down here. So let's do the conversion first. So 0.25 millimeters. Now what are we going to be doing here? We're going to be multiplying, aren't we? To get it up to micrometers. So it's going to be times 1,000. So we're going to multiply that by 1,000. Is going to equal 250 micrometers. Okay, so just remember, as we're going down, we're going to have more of these things. That's the easiest way to think of it. You've got 0.25 there. You're going to have more when you go down a scale, so you're going to be multiplying to get more of them, right? Because the measurements are smaller. There's going to be more. I know that biology is chosen by a lot of students who aren't all that keen on maths. So just think it through logically and you'll get there, okay? And it's going to be a 1,000. You've got your factor here, your conversion factor here. So they give you that. All right. Now what we have to do is we have to get the width of a single palisade mesophyll cell. So it's 250 micrometers right across here. And there's 1, 2, 3, 4, 5. So we're going to divide that by 5. So 250 micrometers divided by 5 is the width of one cell, right? 1, 2, 3, 4, 5 equals the width of one cell. All right, we'll just jot that note in. And that equals, always do it in your calculator just in case, because you never know. 250 divided by 5 equals 50. Now you had that in your head, and the good thing to do here is to make sure that you can make that make sense. So 250 divided by 5 is going to be 50, isn't it? 25 divided by 5 is going to be 5, and you're grand. Okay. Final part of this question is to describe the difference that would be found in the ultrastructure of plant cells that do not carry out photosynthesis. Okay, well, this would be something like a plant cell in the root, where it's not exposed to the light, so there'd be no photosynthesis being carried out, or perhaps in the stem of a tree, in the trunk of a tree, where there's no light gets in through the bark. And the cells of plants that exist in areas where there's no light, they will not have any chloroplasts. Chloroplasts need light to survive, so in any cell of the plant where the chloroplasts can't get any light, the chloroplasts will just die off, or they'll fade away and you won't be able to see them, to tiny little maybe chloroplast spores or something. I'm not quite sure what happens to chloroplasts, but anyway, what you don't see is you don't see them under a light microscope, okay, in the ultrastructure. So describe the difference. No chloroplasts, I think, is all you have to write. That's all you're going to have to write to get the mark. No chloroplasts. Okay. It's a 1 mark question. I don't think you have to. You just describe the difference. You don't have to compare. So you don't have to say the cells in the light have chloroplasts and the cells in the roots would have no chloroplasts, because you're not doing a comparison. You're just describing the difference, and the difference is no chloroplasts. So that should get you the mark. All right. Thanks for watching. I'll see you next time.